Hello, I am going to have to re-record one of the slides because I've made a mistake of not including the slides. So bear with me, and I'll do the recording quickly. Now, before we go further into genetic mapping, I just want to show you this, to share with you this slide that I found online. This is the link up here shows it's Russian, and I went down to I copied that and translated it, and then it says this presentation was published 4 years ago by Elena Veripeva. Right, so wherever you are, thank you, Elena. But because why I'm using this slide is because I think it's got whatever I needed. Okay, so it's on SlideShare. If you want to see the link, wait, how do I do that? Okay, hang on. What have I done? Okay, so it's myshare.ru.slide.1268175. Okay. Now let's go through the slides. Forensics, DNA analysis. Normally you use for paternity suites, victim identification, identifying suspects. So. Usually, identification is based on physical attributes and ethnicity, gender, height, and then you look at friction ridge identification. You know the fingerprint is called friction ridge identification, and then of course you can do blood typing, and so this was all before DNA until Alec Jeffries came up with DNA fingerprinting. This is justification. Between humans, we are 99.9% similar, but when you say 0.1 out of 3 billion is a lot. It's about 3 million base pairs that are still different, that can be used as a telltale identification system. So it says here, pointers, there are still about 6 million base pair different. They're talking about base pairs, and when I say 3 billion is haploid genome. So you can do a fingerprint. Now you may want to read on that. There are two different methods: fingerprinting, profiling. They actually refer to different things. Fingerprinting is one of the first to be introduced by Alec Jeffries. It uses restriction fragment length polymorphism. What is RFLP? RFLP is, well, restriction enzyme cuts certain sequences. Fragment polymorphism is that, between two individuals, one may have the site and the other may not. So that means to say, between me and one of you, say for example, I have the site and you may not. If we cut that particular fragment, then when we run gel, will have different bands than you do. Right, the ones that don't cut will have a longer band. The ones that have that cut have two shorter bands. So you can view that. So so it works like this. So you know Southern blotting, right? You use the probe to detect. Do you know Southern blotting? I hope you do. So how you work Southern blots is that you run your DNA in the gel. Right, all of your DNA, and then you have probes. These probes are labeled with radioactive maybe, and then this radioactive will find its complementary sequence, bind to it, and because it is radioactive, or even if it has a colorimetric system, you can see a color, then you can see where the band is, right? Because it adheres to one location. So this is the probe. This red bit is the probe. This is, let's just say this is DNA from individual 1, this is DNA from individual 2, right? I mentioned, let's say this is my DNA, like this is your DNA, one of you. So in my DNA, there is a site. There are 3 sites there are 3 cutting sites, like it cuts here, it cuts here, it cuts here, but in yours there's a polymorphism, you don't have the middle cutting site. So when I probe, when I use my DNA, run it on a gel and use a probe, because now my fragment has cut there, so the probe is going to bind in that region, so it will detect that small fragment. Compared to yours, you don't have a site there, so this probe will detect a longer fragment, and this is why you get two different bands. Alright, that you need to understand first. Now that is just one fragment. In DNA fingerprinting, you detect many fragments. This is another example. Let's say this individual is homozygous, heterozygous, homozygous without the site. So again, for this individual you will have all short fragments. For this individual you have short fragment and the long fragment. This individual will have or only the long fragment. So you have this kind of patterns. Again, this is just detecting at one locus. What is locus? Locus is location in the chromosome. So you have like 24 different chromosomes. For humans, let's say you have 22, 23 pairs. This is detecting at one site, right? One place alone, one locus. So now, DNA fingerprinting first described in 1985 by Alec Jeffries. So Alec Jeffries as a method to why they were trying to in fact you can read a book. It's called the Blotting, right? Yeah, this book, the Blotting, it tells you of a murder mystery in a town where they actually used the first use of DNA fingerprinting to solve this case. It's quite interesting. I've read the story. Okay, so it was then used to rule out suspect in rape murder in England in 1986. So they used that, and in fingerprints, this is some historical aspect. What is it detecting? It is the probe that they use detects mini satellites. Mini satellites, where are they located? They are located in the telomeric regions of many chromosomes. So that means to say one probe can detect multiple loci. Right? The picture I showed you earlier, it says that it's detecting one locus, one site. But if you are using a probe that detects mini satellites occur on various chromosomes, correct? So that means you have one probe, it will bind to many different places. That means to say, if you have your DNA, right? You have your DNA and then you digest, you make a precision digest and you run them on the gel, you use the mini satellite as a probe, you are bound to get many bands, correct? Right? Again, that is a fact, digest that fact. Okay, so this two slide talks about the repeated units, the source of variability. So I just mentioned in fingerprinting they use mini satellites. In DNA profiling later they will use microsatellite. Microsatellites, the difference is mini satellites, the core repeat unit is about I think 30 bases being repeated on and on and on. But for microsatellite, the core unit is smaller. In this case it's 80, or CATG. So it's like if it's a repeat of two bases, it's called a dinucleotide repeat. If there is a repeat is based on 4 bases, you call it a tetranucleotide repeat, things like that. So that's microsatellite for profiling. Mini satellite is for fingerprinting. So because the length, the variation between you and me is of the length, right? And because these are repeated side by side, they are called tandem repeats. And let's say for a particular locus, I may have 4 repeats, you may have 6, another person may have 8, another person may have 2, or a 7, so that's why they call it variable number of tandem repeats, VNTR, right? Certain locus is so variable, that means in an individual, in a class, for example, of about 20 individuals, everybody has different. So this is what we call as hyper variable. Some are not very variable, between in a class of 20 individuals, you'll find that there are only about 4 variations. So that's not very variable. But there are some that you look at 20 different people, you still have 20 different lengths. So that is very, very hyper variable. Okay, all right. So this is the variability. Where do you get variability? This is an example of unequal crossing over, because when chromosomes cross over, what they do is they pair, right? They will pair, they will align, and then they pair. And then the pairing is based on sequences. But if the sequence, in this case, right, each box represent a particular sequence and repeated and so on. So when they align, they can align here, they can align there, they can align there, because the sequences are repeated. So they make mistakes in alignment, and because of that, if there is a crossing over, then you'll get the product of the crossing over. Although originally they are both the same length, what you get is one is longer than the other. Okay, this is another concept that you have to digest. Okay, southern blotting can be used to visualize variation. Again, let me remind you again, we're talking about DNA fingerprinting, and these are the probes, the 2 common probes, and this is multi-locus mini satellite. Remember I mentioned, because mini satellite occurs at various chromosomes, so using one probe, you run it on a gel, you'll get multiple bands. This is an example of how it looks like. One locus, right? One locus. This is one locus. This is the variability. One person may have 1 repeat, the other person has 2 repeat, the other one has 3 repeat, 4 repeats, and so on. The rest of the sequence are the same. The difference is just between the repeat units, and because of that, you will see variable length of one locus. So this is a typical example of a fingerprint, right? Here you have a blood stain, right? This is a blood stain. You do a fingerprint, and then you get this type of bands. And from here, it's quite. You start comparing with the present and absence of these are all the different samples, and you will see immediately that well, for classroom purposes, you can see definitely that this one matches, right? This is the blood stain, that one matches, so this will give you a match. This, and the person who has this fingerprint, you can almost be sure that that is his blood stain, right? So this is how it works. You do that, you don't look at a particular band, you just look at the overall present and absence of. See, for example, that band is present there as well. That those two share a band and so on. So if you look at multiple bands, and for this one, one match, 2 matches, 3 matches, 4 matches. You normally look at the dark bands. 5 matches, 6 matches, 7 matches, 9 matches. So if it matches a lot, then it increases the probability that these two are from the same individual. Okay. This slide says that they want to show. You remember Dolly, the sheep that they cloned. They want to show that the clone is indeed from the original parent. They did fingerprinting. So these are the other cells. This is from the cell culture. This is from the dolly. So they find that from other cells, cell culture, dolly, they have the same pattern, and these are all different sheep. And it's just to show that fingerprinting is quite specific for the individual alone, because you're talking about multiple bands. Chances people can share 1 band, 2 bands, but chances that different individuals share all the bands together, well, it's only limited to identical twins. Next, so okay, now again, the issue with fingerprinting is that you're looking at multiple locus at the same time, and then you get this kind of results, and it's not easy to interpret. Then comes DNA profiling, and here you look at individual locus. So that means to say you zoom in on one site, so you get back, you get this kind of result. It's either you're homozygous or heterozygous or homozygous on the other allele. So this is one locus. So because you use probes that are only detecting one region. Okay. So this is an example. All right. So one mother, child. This is the alleged father. You can see that if that is the mother, if that is the child, now that band must come from the father. This is called the obligate pattern, right? Must. If you want to know, if you want to tell whether one person is the father or not, they must have that band. And so this is, if this is the alleged father, you can say no, it cannot be, because the father's band do not appear here. But this one, control, what is a C? So this one is just want to show that you mix the child and an alleged father. Now you can see 1, 2, 3, 4 bands. So it doesn't. But unlike this one, you see this is another alleged father. So you have, again, this is mother, this is child, this is child. So that means that one must come from the father. And in this case, the father has the same bands. And when you mix the child and alleged father's DNA, you are going to just see one band here, one band here, and one band. It's not like this. So this is taken as a match. This is taken as a mismatch. All right. So it's easier to see because you're now just looking at one locus. Okay. Okay. Here you see one band, right? One band, one band, one band. But here you see multiple band. In actual fact, when you do this in the laboratory, you'll get this kind of results. But you know, the only band that matters here is the dark ones. Why you see many bands? This is what you call as the Stutter Effect. I don't know whether you can see here. Let's try and Google Stutter Effect gels images. Yeah. If you see something like this, you know, you want to see the band, but you see other company band that's not as bright. Stutters, you know, it's just something that you see when you do this kind of study, right? So you see stutter bands, right? This is the main band, but those are the stutter bands. Those are, oh sorry, these are these are the ladder. But basically sometimes it's also called the shadow bands, right? But never mind. So FRI femur was extracted from the femur was compared to the son R and wife I. Okay, at 10 loci to found fully compatible paternity. This one is a case where they exhumed Joseph Mengele, and then they did some profiling of the corpse skeletal remains to prove that whether this is the husband or not. So this is what they did. And so of course this one shows that, yeah, this is the son, this is the mother, and this is the father. So there are alleles that are being shared, so that supports the evidence. So rather than use probing and southern blotting, now people use PCR amplification. right? So the U typing. And so basically what you have primers that is designed to amplify from the flanking region. This is the repeat unit, so your primers will bind there. Your primers will bind there, right? These are the unique sequences, right? They will bind the same place, but because one person has longer repeats than the other, then you get two different bending pattern sizes. The sizes are determined by the number of repeat units. So look at this. You have three individuals, individual B, C, F. Now what it's trying to say is that, okay, where is the match? Different colors refer to different locus. Okay, so for this locus, A is that and that, B is the same, C is different, and F is also the same. Okay, what about if you look at the green locus? The green locus for A, it's there, for B is there, for C is there, and F is there. And then for the blue locus, A is there, B is there, C is there. Now you see from this, right, if you're trying to determine who or where, if there's a match between F and either A, B, or C, you'll see that for one locus, right, for the red locus, A matches and B matches as well, right? But if you go further, if you look at the green, the blue one, blue matches, but A does not match anymore. And then the green one, B matches, again A does not match. So there is a chance that there is a match between individuals because you're just looking at one locus, right? If the locus is not variable, as I mentioned, some locus is very hyper variable, some is not. Some, I mentioned earlier, you see in a class of 20 individuals, you know, you look at one locus, everybody has got different ones, but some, you know, maybe you can see that, oh, at least 5% share one allele, another 5 or 6% share another allele, so that's not very variable. So this happens in markers. So in this case, there are three VNTRs. You have VNTR1, there is a match between A and B, but if you go further, if you look at the green and blue, then only B matches completely with F. What's important from this slide is that to show that the chances of having a match, there is a chance that for individual locus you find a match within individuals, but the chances of getting a match will be reduced if you look at a bigger number of loci, right? So the more locus you look, the chances of getting exact match reduces unless that's the same sample, right? So this has been the basis of paternity testing, right? This is an example of a DNA sequence where you can see, if you read sequence and so on, you'll see that but this red represents the VNTR, variable number of terminals. You see AT, AG, AT, AG, AT, AG, AT, AG, AT, AG, you know, that's just one locus. So if this picture says, okay, you're looking at three different VNTRs, the standard for paternity tests, if you look at you know, criminal cases and so on, when you want to do identification, the minimum number of locus you use is 13 core loci of tetrameric leaves are tested together to make a DNA profile. So you're looking at 13 different places. In fact, nowadays for certain laboratories, for certain applications, you're not looking at 13, you're looking at 21 different lockers. So initially, so there are different systems that you can see depending on what you need it for. All right. Primers have been developed to allow amplification. This is an example. Previously, during my time when I studied, I deal with a lot of this gel and bands, and you get your eyes twisted looking at them. But nowadays, you have computers to help you out. But instead of looking at bands, you're looking at peaks. Okay, this is one locus, right? So you have. Okay, this is now. Let me just try to explain this. Remember, it says 13 core loci. Sorry. 13 core loci. This is from a marker. This is the name of the marker. On another slide, I have the list of the different locus that's used for identification. Examples include this. D3S1358 is one locus. VWA is another locus. FGA is different locus. And it says that, okay, for D3S1358, they test a different number of populations, and then they say that these are all the different alleles that is present in the population. For VWA, these are the different number of alleles. This FGA, you have different number of alleles. These are different alleles. And for this individual, for that locus, for that marker, is a homozygous 15. For VWA, is heterozygous 14, 16. These are alleles. FGA is 24, 25. So this is the genotype for this individual. So you imagine this is just 3. You actually test for 13 or maybe up to 20, to give an individual a genotype, and then you compare between different samples. This is another example. So D13, 15. So this is the genotype. VWA 16, 16. FGA 19, 24. These are all the locus. These are the genotypes. This frequency is the number that tells you the rarity. Genotype 1616, it says in the population you'll see this in 4.4% of individuals. They must have done this some study. Before you can do anything, you have to do a study to see the population frequency. Say for example the TH01, the alleles are 9.3, and this occurs 9.6% of the time, right? So example of DNA profile, just in 13 CODIS. CODIS is a combined database of combined database of identification system, something like that, right? This is used by the FBI. I'll show you in a little bit and what this thing are. And the odds of another person having the odds. Okay, it's not very clear here, but it says the odds of another person having this profile is 1 in 7.7 times 10 to the power of 15. Now that's the odds. If you look at 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, there are 14. This STR, there are 13. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So the chances of another person matching the same genotypes are actually 1 in 7.7 times 10 to the power of 15. This ML is melogenin. This is used for gender identification, sex identification. So X and Y. So this person is a male. Okay. We'll go this later when you want to do parentage testing. Okay, there you go. CODIS is combined DNA index system. And as every day I announce a selection of 13 STR loci. Okay. STR alleles are easily genotyped, so they are kits to help you genotype. So this is the they see it's in X and Y, the amylogenin. D21 is on chromosome 21, D18 is D60. Why they chose this? Because these are the best lockers, best in terms of they are very variable. When you are something that is very variable, right? For example, if you look at this one, it says, for FGA, for example, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 different alleles in a population. Chances of getting a match is, or by chance to get a match is reduced. All right. So this lecture focuses on DNA identification. Because why is this important? Because later on you want to use this in genetic mapping. Okay. All right. So have a look at this and bear this in mind, so that later on we can use the same principles in genetic mapping.